So thank you for having me. This is a beautiful city, my first time. So I'm very pleased to present to you and happy that you selected this abstract. This is an update on bb2121, which is a promising therapy for heavily pretreated relapsed refractory multiple myeloma patients. These are my disclosures. I play with everybody, so I'm not partial to anyone, as you can see. I guess I got to keep it up there for a little bit. So, multiple myeloma, as you all know, is the second most common blood cancer, and we've had great advances in myeloma over the last decade, and there's many approved therapies, and patient survival has improved, but unfortunately, despite all this remains an incurable disease. And more sobering is, as patients use some of these therapies, the more common being the proteasome inhibitors and the immune modulating therapies. As patients become refractory to these and have had more than 3 prior therapies, their overall survival is only 8 months. B-cell maturation antigen belongs to the tumor necrosis factor receptor superfamily. It is nearly universally expressed in multiple myeloma cells and a few mature B cells, so it makes it a great target to be very specific to multiple myeloma. And bb2121 is a second-generation chimeric antigen receptor, or CAR, T cell that targets BCMA and induces a very rapid and sustained response in patients with heavily pretreated relapsed refractory myeloma. This is a busy slide, but it's actually relatively simple. On the top is what the patient actually goes through. So the patient is screened. Once they meet criteria, they are leukapheresed. This is how we collect their T cells. The T cells are then sent out for manufacturing of the bb2121 product. And this takes about 10 days, but then you have to have all the releases, make sure it's not infected, etc., and that can take another week or so. So it's usually about a 3 to 4 week process. Once the CAR Ts are made, the patient is then given what we call lymphodepleting chemotherapy, fludarabine Cytoxan, which is a low dose of chemotherapy to try and basically quell their own T cells so that they don't compete with the CAR T cells. The CAR Ts are then infused on day 0, and that is the only treatment the patient gets. They're in the hospital for observation of toxicities, and they then get released from the hospital, and the first assessment is at month 1. In the dose escalation, we had 21 patients that were treated with dose levels, as you see there, 50 million, 150, 450, and 800 million cells. And in the dose expansion, we had 22 patients, and we treated 10 patients at doses, 22 patients at doses between 150 and 450. These are the results. In terms of toxicities, there are 2 main toxicities that you worry about with CAR T cells. One is cytokine release syndrome, which is not too dissimilar from sepsis, and the other is neurotoxicity. And this has been described well in other CAR T programs. As you can see, we saw 63% of our patients had cytokine release syndrome, and 33% had neurotoxicity. But most of these were grade 1 and 2, which are sort of the lower grade. So most people just had a fever. Some people had very transient low blood pressures that resolved with some fluids. And then only 5% and 2% of our patients had grade 3 or greater cytokine release syndrome neurotoxicity. Most of the patients had drop in their blood counts, as is expected. Most recovered by day 32. We had no grade 4 CRS events, and we had no fatal CRS or neurotoxicity events. These are the responses. So as you can see, there's an actually dose response curve. So at 50 times 10 to the 6, this is what we consider a subtherapeutic dose level. We had 1 transient response, and then at 150 times 10 to the 6, we saw an overall response rate of 57.1%, and patients who received more than 150 million CAR T cells had an overall response rate of 95.5%. These patients did not show you had a median prior therapy of 7 to 8 prior therapies, so very refractory, heavily pretreated patients. In terms of the tumor response, there's a question of whether you need a certain amount of BCMA expression to be able to respond to CAR T cells. And as you can see on the right panel, patients at the 450 million CAR T level, there appeared to be no difference in response, whether you were BCMA low or BCMA high. Our median progression-free survival in the 18 patients in the dose escalation, which are the ones who are furthest out, is 11.8 months. And in patients who achieve MRD negativity, and this is something, it's a much deeper level of response that we are now evaluating in patients in the frontline setting and early relapse. It's never been tried in this very heavily refractory population. So the fact that we can even reach it is impressive. So 16 patients were tested and 16 patients were MRD negative, and in those patients, the median progression-free survival is 17.7 months. So in conclusion, bb2121 and active doses of greater than 150 million CAR T cells induces deep and durable responses in heavily pretreated populations in the relapsed refractory setting, myeloma. Median progression-free survival 11.8 months. 100% MRD negativity in the 16 evaluable patients with a median progression-free survival of 17.7 months. And I'll just put this in context in that the two last therapies that were approved in multiple myeloma in patients who had more than 3 prior therapies had median progression-free survivals for 3.9 months. So this is actually quite an impressive result if you put it in that context. To date, the safety profile of bb2121 has been very manageable at doses as high as 800 million CAR T cells, mostly grade 1-2 CRS events with infrequent tocilizumab and corticosteroid use, which are the drugs we use when these toxicities become more higher grade. We had one case of reversible grade 4 neurotoxicity, and we have not seen others in the drugs in the dose expansion. So the next steps are the bb2121 is now open in a global pivotal trial called KARMA. This trial is open for enrollment in North America and Europe, and then additional trials are planned in earlier lines of myeloma, including frontline therapy in high-risk patients.